Hello, I'm Mark Newman, Chief Scientific Officer of GeoVax. Today's presentation is on our program to design a universal SARS-CoV-2 vaccine, which will be effective against evolving variants. For this presentation, I'm going to introduce you first to the immune responses to the existing SARS-CoV-2 pandemic. This is data generated in other labs that we're using to direct our design. Then I'm going to introduce you to the conserved regions of coronavirus and why we think that this is be a valuable approach to designing a vaccine, looking at inducing immune responses to conserved areas, areas that are less likely to mutate and evolve over time. Then I'll introduce you to the GeoVax modified Vaccine-Virus MVA-VLP technology. Our vaccines are based on the use of MVA, which supports encoding numerous viral target proteins. And in this case, we are including sufficient proteins from CoV-2 to support virus-like particle production in vivo. Finally, I will show you some preliminary data on the MVA-VLP COVID vaccine, basically the prototype product that we're working with. Immune responses to viral infections are commonly characterized as being antibody-based, which are humoral immune responses, and cellular immune responses, T-cell mediated immune responses. So antibodies will be specific for viral proteins. They block the virus from infecting or can block cell-to-cell spread mediated through viral proteins. Cellular immune responses directly limit viral replication, typically through the action of cytokines, but they can also clear virus-infected cells through direct killing. An optimal vaccine will induce a multifunctional immune response, meaning that it will induce both cellular and antibody responses, and it will induce memory responses, supporting the recall of both cellular and antibody responses upon subsequent challenge. Characterization of SARS-CoV-2 antibody responses has been completed in a number of labs, focusing on serum obtained from individuals that have recovered from infection. So antibodies specific to the spike protein are present in most serum samples obtained from patients. Many of these will neutralize or block infection of the CoV-2 virus. Neutralizing antibodies will persist. At this stage of the game, we know for months. Long-term data is still absent. CoV-2 infection typically does induce bone marrow resident plasma B cells in humans. These would be the source of memory cells. So an active infection will induce some level of immunological memory in humans. Data generated in multiple laboratories suggest that the duration of neutralizing antibodies, those specific to the S protein, may be relatively short-lived in COVID-2 infections. This is actually consistent with antibodies generated against the seasonal coronaviruses. This indicates that a vaccine may require repeat doses, potentially yearly, similar to the influenza virus vaccine market. Non-protective antibodies which actually are either low in titer or recognize proteins other than the S protein, have been associated with antibody-mediated enhancement of disease in certain animal models. Luckily, this doesn't appear to be a case in SARS-CoV-2. However, focusing the antibody response correctly remains a concern. Now, the stabilized S protein is the antigenic target, which is the focus of most of the first-generation vaccines. These are the products that are produced and approved by Moderna, Pfizer, J&J, AstraZeneca, and Novavax. So these products are designed specifically to induce neutralizing antibodies specific to the S protein. SARS-CoV-2 is an RNA virus, and like all RNA viruses, it's subject to mutation. Mutations can accumulate over time. These can actually evolve into stable variants within the population. A variant might be more transmissible, or it might be resistant to prior immune responses generated through either vaccination or through prior infection. The illustration on the right demonstrates how complex SARS-CoV-2 circulating population has become in a single year. Each of these represents a related virus that is mutated in some form or another. This is a concern because if mutations accumulate in critical proteins, for example, the S protein, you might generate a variant that is not impacted by the vaccines, resulting in immune escape. This is a limitation commonly associated with vaccines based on just a single protein. The reduction in vaccine efficacy, measured as a function of antibody activity, is demonstrated in these slides. Here, antibodies raised against the Moderna and Pfizer mRNA vaccines, which are specific to the spike protein, are measured in a virus neutralization assay. Antibodies generated against the vaccine are very effective in inducing the parental variant, but over time, variants that are arising with the pandemic are more and more resistant to neutralization by these antibodies. It's a clear indication that the virus is capable of generating variants which will essentially escape the activity of neutralizing antibodies induced by vaccination. As I showed in a previous slide, a number of variants or mutations are accumulating within the SARS-CoV-2 circulating population. A number of these are leading to stable variants that are in fact demonstrating increased infectivity, transmissibility, and potentially decreased or increased resistance against antibodies, both the vaccines and the monoclonals. I'm not going to go through all the data on these particular variants, but 8 variants have been identified as variants of WHO concern. These all represent potentially more virulent forms of SARS-CoV-2 in that they either have an increased infection rate due to mutations within the S protein that allow for tighter binding to the receptor, or they are more resistant to the activity of neutralizing antibodies. This includes antibodies generated against vaccines and some of the monoclonal antibodies that have been produced. Multiple laboratories are also investigating cellular immune responses against CoV-2. Over 90% of convalescent patients have detectable CD4 T cell responses. These responses are multispecific, often indicating both functionality and memory. They'll produce cytokines or direct activity against the virus. Multispecific and multifunctional T cells are associated with accelerated viral clearance and with an increased level of protection from severe COVID. Patients who recover from SARS-CoV-1, the original SARS virus, possess long-lasting memory T cells up to 17 years after the outbreak. This indicates that a recall response can be generated with CoV-1, and a vaccine that can induce a similar recall response against CoV-2 would have some clear advantages. T cells recognize epitopes that are expressed or contained within multiple proteins. This includes both the structural proteins and the non-structural proteins. One observation is very interesting in that T cells from uninfected donors recognized epitopes in the CoV-2 non-structural proteins 7 and 13. What this suggests is that cross-reactive recognition by T cells that were induced against a seasonal coronavirus and are now recognizing the same highly conserved epitope within CoV-2. Conservation of T cell epitopes can be quite extensive. For example, the non-structural protein 7 T cell epitopes are conserved widely against animal beta coronaviruses. This provides an opportunity to design and produce vaccines against epitopes that are conserved. If they're conserved within multiple coronaviruses, this indicates they're not susceptible to variation and thus unlikely to be the basis for forming escape mutations. As mentioned previously, vaccine induction of T cells is likely to be required for optimal protection, in particular long-term efficacy based on observations of long-term memory. If we look at the possibility of emerging variants where the variation within S proteins is impacting neutralizing antibody effectiveness, then the T cell responses to conserved epitopes provide the basis for the long-term protection. Other data that suggest the importance of T cells are related to disease severity. What we've seen is that functional T cells are really well correlated with reduced severity of disease, maybe even more so than circulating antibodies. This is what this data shows, that multifunctional and multispecific T cells, if they're detected at a high level in patients that were infected, documented infection, but in fact did not suffer severe disease. Whereas patients with severe disease can have T cell responses and high levels of antibody responses, indicating that while the antibody responses limit the initial infection, they don't do much to protect an individual from progressing to disease once infected. This is another important component of the T cell response. Bear in mind that the goal of vaccines is always to stop severe disease. We know that we're never going to totally protect with sterilizing immunity infection of individuals, but success can be mediated by controlling disease, keeping people out of the hospital. Now, to design a vaccine that induces immune responses that are not impacted by variation that is accumulating within the pandemic, we need to target conserved epitopes. So these are epitopes that are present in all sorts of related coronaviruses, not just different variants within CoV-2. If we look at multiple coronaviruses, we find that the spike protein, the S, open reading frame 3 and open reading frame 8, actually are somewhat variable. As we've seen in the CoV-2 situation, variation within the S protein seems to be leading to resistance against antibody responses. So to target a vaccine or to design a vaccine that is impervious or not impacted by the variation that is surely going to accumulate, we need to target these conserved epitopes. And the epitopes that are most likely to be conserved appear to be within the non-structural proteins, open reading frame 1A and 1B. As I mentioned previously, the observation that T cell responses can be detected in individuals who have not been infected with CoV-2 indicates a cross-reactivity, most likely between seasonal coronaviruses and CoV-2. These would be conserved T cell epitopes, and in fact, these do exist. These data show that different epitopes identified in the open reading frame 1A, 1B non-structural proteins are in fact immunogenic and recognized by T cells from both exposed and recovered and unexposed individuals. These represent cross-reactive responses, but are really ideally suited to inducing or designing a vaccine that would not be impacted by variation, for example, within the S protein or some of the other non-structural proteins. So how do we design a vaccine that will induce both antibody and T cell responses, in particular responses to non-variable COVID gene products? So what we're working on is the use of the modified vaccinia virus vaccine vector. The reason for working with MVA is it has a large coding capacity and multiple cloning sites, which allow us to encode multiple CoV-2 proteins. So in this diagram, what this represents is, in fact, we can encode antigen, the blue antigen, the orange antigen, and the green antigen. In this case, it would be the S protein as an antibody target, the membrane protein, and the envelope protein, which provides structural basis to form virus-like particles. This is the kind of core component of the GeoVax MVA-VLP vaccine platform. That is to use MVA to encode an antibody target, but also structural proteins that support the in vivo production of virus-like particles. Now, why virus-like particles? The VLPs present the antigen in the most native manner that we can use. They're not single proteins. They would be lipid envelope based and presented. They'll be concentrated on a particle. We would believe that these are basically the most authentic way of presenting the viral antigens. Now at GeoVax, we've designed several prototype vaccines encoding multiple CoV-2 proteins. What I'm showing you here is the data around the lead candidate, referred to as CMO2. This MVA encodes a stabilized spike protein, the membrane protein, and an envelope protein. In the lower left-hand corner, you can see one of the critical aspects of this. And in fact, MVA-infected cells do produce virus-like particles which will bud from the surface of the cells. This is a very stable virus, and that we've undergone 20 passages within cell culture, and the insert is still stable. The vaccine, the MVA, is still producing the needed proteins. We can demonstrate that this CMO2 produces protein that is identified within cell supernatants, indicating the secretion of the virus-like protein or the secretion of the VLP, and we can detect membrane by immunohistochemistry staining of cells that are infected with the MVA. So at this stage of the game, we've been able to document that all the critical aspects needed for this vector to be used as a prototype vaccine. Now, to test the efficacy of these prototype vaccines, we're using two models. Here I'm showing you data in the hamster model. This is designed to actually document protection or diminution of disease and recovery from infection. What you can see on the left-hand slide is that the MVA vectored vaccine will induce antibody responses measured by ELISA in all of the animals after a single dose. This is boostable with the second dose, as you can see, on day 56. Now, importantly, if we infect these animals, we do see an initial infection suggesting initial infection documented by a slight loss in body weight, but animals quickly recover. And there's a significant difference between animals that have been vaccinated versus those that have not. Now, we are also using the human transgenic ACE2 mouse model to evaluate immunogenicity and protection. Here's preliminary data prior to the actual infection, which we haven't yet done. What this shows on the left side is that a single vaccine will induce significant neutralizing antibody responses. On the right side, we're showing that these vaccines, either a single or a prime and a boost vaccine, will induce both CD4 and CD8 responses. We're getting immune responses not only to the S-peptide pool, but also to variant peptide pools and to the membrane pool. So this indicates that CD4 and CD8 responses are in fact induced to the dominant target, the S protein, but also the membrane, and they're recognizing variant forms of the S protein. We're also seeing T effector memory and T central memory induced, which is pointing to the fact that we're likely to be inducing potent memory responses. Now, these animals are to be challenged at the time of this presentation, but we'll be generating actual efficacy data shortly against multiple variants of CoV-2. So how are we interpreting this data? Well, the first point I want to make is that we believe this documents that the MVA vector supports the design and production of next-generation vaccines. These are vaccines that will encode multiple viral proteins. The current generation product that we're working with encodes the S protein as the dominant neutralizing antibody target, but also the M and E proteins as potential T cell targets. And in this initial data, we've already demonstrated that the M protein and the S protein both induce cytotoxic CD8 and CD4 T cell responses. Now, the combination of the S, M, and E proteins within a single vector supports the VLP production within the cells. This is important because, as we said, believe this will optimize the immunogenicity. The S protein will be expressed and presented to the immune system in a particulate, also within the lipid membrane milieu that is normally seen in a viral replication. Functional T cell and functional antibody responses have been induced. Functional antibodies in terms of virus neutralization, and T cell responses including T cell effector memory and central memory. These indicate that the vaccine should induce immune responses that will mediate for protection against infection, but also against pathogenesis, which is demonstrated in the hamster model. Now, to produce vaccines that induce truly universal protection against evolving or emerging variants of CoV-2 and potentially against other beta coronaviruses will likely require that we induce or increase the breadth of the T cell responses. As I've shown you, data in humans indicate that individuals that have not been infected with CoV-2 have T cells that recognize epitopes in non-structural and some structural proteins of CoV-2. There's a significant immunological cross-reactivity between coronaviruses that are capable of infecting humans. So the next goal here is to build upon the existing CMO2 MVA construct and add to this individual proteins or genes encoding individual proteins from the non-structural component of the virus. There's a lot to choose from here. Over 60% of the viral genome encodes these non-structural proteins. We know that they are sequence conserved, and we know that they're immunogenic in humans. So the next generation of vaccines will include non-structural proteins under different promoters that will support the endogenous production within an MVA-infected cell and the generation of CD4 and CD8 T cells, and the VLP structure that will present the S protein within a VLP structure to induce antibody responses. With that, I want to acknowledge key members of the GeoVax team and the University of Texas Medical Branch, and of course, funding by the NIH.